So a lot of you know me, a lot of you don't. I started this as a federal forum and it started with the Navy. We branched out into all the federal government. The idea behind this is that innovation has been stifled by one thing where we try to stovepipe it. We take people that are subject matter experts, been doing the same thing for the last 10 to 30 years, and we put them in a room and say, hey, figure out how to do this different. Coming from a lean Six Sigma background, I chose to apply those principles for process improvement to innovation. And one of the main principles of that is to have people that don't understand the process from outside the process evaluate the process. Those people are going to ask the questions, they're going to want to understand. And during that interaction with the people that do run the processes, that's where you have your aha moments. That's what we're hoping to create here. This is a very fast paced. The presenters are instructed to discuss what it is they do, why they do it, where it applies, and then we want you to understand and see if this actually applies in your industry. There's a lot of applications, cross industry, cross sector. One of the stories I like to tell, and I'll share it really quick here, is would you think about discussing or contacting a marine biologist to help you solve an aerodynamics problem? Most people would say no. I ask the question, why not? Because aerodynamics is closely related to hydrodynamics, and hydrodynamics is fish. We had a presentation by a biologist studying fish surfaces that generated well over a dozen collaborations within the shipbuilding industry as well as aeronautics. So that's the goal here. With all of that, I will turn this over to Biosortia and their fascinating technology that they've put together. Ross has a lot of good things to talk about. And just for Ross, you may want to start out slow because people are coming in like crazy.
Terrific. Well, I appreciate that, Eugene. You hear me just fine, I take it?
I do.
Excellent. So what we're going to talk about today is industrial scale microbiome mining and collaborative innovation possibilities around materials, defense, and energy. Now, next session on February 28th and then on March 27th, we're going to talk about therapeutics, health, and nutrition, then sustainability, environment, and agriculture. Now, how could one technology potentially encompass so many different areas and opportunities? I hope with this presentation today, it kickstarts the thought process of how we could work together, because what we've got is we've got access to what is essentially the dark matter of biology, the hidden cellular signaling, which any kind of life science area truly depends upon. So we're going to talk about that. We're going to provide a little background, then we'll get specifically into the technology and into what that technology could potentially impact related to today's topic. So let's go back a little ways. Let's go back to the formation of this planet, this solar system. And shortly after the start of Earth as a glowing hot mess in this solar system, about 4.5 billion years ago, there is a belief that less than 100 million years after the Earth truly formed, that there could have been the start of the first microbial life on this planet. Now, that can't be proved because the records have been basically destroyed, so there is no fossil record to see. But essentially, once life was started on this planet, it terraformed the planet. It basically turned it into the world we see today, but that took 3 billion years. Because basically 4 billion years ago, it was a soup of hydrocarbons with no atmosphere, and those microbes started the first life. Ultimately, it evolved into all life itself. So when you think about every organism that has ever lived, their origins are microbial. Now, when we look at today, we're learning that microbial life actually is involved in every organism today as well. So let's take, for instance, just the human and the microbial origin, not only of our species, but of the microbes that live in us. So right now we only have 30 trillion human cells, but in each one of us is approximately 40 trillion microbes. So we're currently outnumbered by the microbes that live in and on us. Now, we've all heard of 23andMe, which is the 23,000 genes that the human has. Well, believe it or not, that microbiome, those microbes that live in and on you, more than 25 million genes. Now, how does this relate to all other microbes and all other microbiomes? Well, a lot of the human microbiome is environmentally sourced. So we could take something like the aquatic environment and find that 73% of the genes and the metabolic pathways of the ocean overlaps our own human gut microbiome. And for those that have not been following science over the last 10 years, what we are learning about the involvement of our human microbiome in our own health and welfare is phenomenal. And that's part of the message here, is that this hidden dark matter of biology is virtually completely unknown. If you went to the Human Metabolome database and searched small molecules or metabolites that are known from the human microbiome, there's 144 molecules listed. But yet our microbes are making over 100,000 plus small molecules that enter our circulatory system and impact our health, which means the vast majority of what's impacting the human is actually invisible to research. And that's a challenge. And that's not a challenge just across human health, but across all sorts of life sciences itself. So let's talk about microbes have been part of virtually every food chain. We're familiar with how microbes impact cheese and how microbes impact other products like beer and wine and bread and things like that. But the reality of it is without microbes, there would not even be food sources on the planet. So again, when we think about all life forms, we've got to be able to connect them to microbes themselves, not just microbes and viruses are bad and we need to kill them, but they're actually an essential component doing good things in the world of life sciences, in the world of biology. So when we talk about the power of microbes, rapid evolution has occurred in microbes, not only through the vertical parent to child type of transfer of genes, but the horizontal transfer of genes. One of the things that the genomics revolution has taught science is that we have not only our genomics we are created with, but as we live, our genomics change. One of those big factors for changing genomics or epigenomics is the fact that microbes that live in and on us interact with our own DNA as we age. So when we start to think about the power of microbes, the transfer of the genes, it's almost nearly incomprehensible. To put it in a little bit of perspective, one species of microbe, E. coli, which I would imagine most of us have heard of, we think of it, we don't want it in the food we eat, but yet it grows inside of us as one of the largest population of microbes that are in the human body, and that's E. coli. But if you looked at the E. coli on this planet and you said, what is its calculating power just based on its mutation possibilities, what it mutates on average according to science, you would need to match the fastest computers, fill up the entire state of South Carolina with that fastest supercomputer in order to potentially keep up with E. coli's calculating power. Now, that's not feasible. And in fact, in some calculations, it would take the entire size of Texas to do the same thing. So when we think about trying to understand biology, some of this is much more complicated than is even conceived today with the fastest computers possible. Now, we're going to use an example right here of how fast the calculating power is, if I can get my slide to work. And Eugene was right. We probably should have tested this. It might be trying to load, so I should give it a second to load.
All right, if anybody has any questions, be a good time.
Well, I am not going to delay on this slide. I'm going to describe this slide. This is from a Harvard research group, and we will make a link to this slide available after this presentation. What they did is they made a very large agar plate, basically a growth system for microbe. And in this growth system itself, what they did is they put different segmentations of 1 times a antibiotic, 10 times an antibiotic, 100 times the power of an antibiotic, and 1,000 times the concentration of an antibiotic. And what this 2-minute video shows is in an 11-day period, it overcomes the 1 time the antibiotic, the 10 times, the 100 times, and breaks into the 1,000 times the concentration of the antibiotic, showing that essentially the microbe itself was mutating as it ran into a problem and figured out a solution. And it's a very impressive Harvard presentation, so we'll make that available to you. And I apologize that it didn't work this time. So we'll move on to the next slide, I believe. There we go. So one of the interesting things is microbes are everywhere and they seem to be able to grow everywhere. So it would seem like we would be able to get at microbes in endless supplies in growth systems or in culturing. The odd part is that growing microbes in a lab is hard. Less than 1% of the microbes on the planet easily grow in a lab. And most of the information we know about microbes have occurred in a lab. But one of the challenges is that just because you put a microbe in a culture does not mean it's going to produce any of the interesting metabolic pathways that it would do in the wild or the wild habitats that microbes live. So the challenge we run into is how do we really understand microbes that we can't grow, which is the vast majority. The other thing we'll point out is that less than 1% of the microbes we could grow has directly, indirectly, or inspirationally led to more than 50% of the drugs on the shelf today. That is an important concept because the question comes up, what will happen if we truly have access to that other 99%? So over the last 50 years, they certainly tried culturing and it showed phenomenal successes, but then they ran into the wall of finding the same chemistry and finding the same organisms and the same metabolic pathways. It wasn't until the genomics era that they really realized that there were millions upon millions, potentially even billions. And in some cases, there's the belief that it could be more than 1 trillion species are not culturable. So we've used things like combinatorial chemistry to try to make nature's chemistry. And the problem with that is there were too many options. So decades of trying combinatorial chemistry, billions of dollars a year, only led to one drug on the shelf. Ultimately, all the indirect technologies, such as genomics, synthetic biology, other omics possibilities, have been a pathway, but they're not really getting to the chemistry itself, and certainly not easy. So just in the drug discovery side of the equation, what we've seen is a decline in new molecular entities that have actually been approved by the FDA, while the cost have climbed 84% faster than inflation. So culturing didn't really work. Combinatorial chemistry really didn't work, at least as getting at everything. Certainly culturing worked, but it left so much uncovered. And then the omics era is a great tool. It's certainly enlightening a lot, but it is not bringing us to the solutions that we seek in life sciences. So the real question comes up is, can there be another approach to get these microbes? They exist everywhere. What if we go get them from the natural world itself? Well, they are everywhere. They're on the door handle of your door. They're on your desktop. They're on your pen and pencil. but they're in such small quantities you can't obtain them. And ultimately, if you did try to obtain them, they're so small that no matter how you try to process them with traditional technologies, they blind filters nearly immediately, which means you can't get much. And if you put too much pressure on it, then you're going to break the cells and lose those valuable metabolites and molecules. And if you wait too long after you've captured it, they start to degrade within minutes. So there's a challenge of even capturing microbes themselves. But where do microbes live in stable quantities? Let's take the Chesapeake Bay, for example. Within that is numerous habitats where there is a stable supply in quantity of microbiomes. And like I mentioned earlier, all microbiomes are interconnected through the genes, the metabolic pathways. They may not be 100% match, but there's substantial matching and overlap of that production of materials. So we would look at something like the Chesapeake Bay and say that is a tremendous source of a stable microbiome for a period of time. Its stability will always continually change, but if you're looking for a large sample, that would be the start. Now you have to have the technology to get that sample. So what we're going to talk about now, because we already talked the reasons why microbes matter. They have produced a lot of products. They have the opportunity to solve a lot of our problems moving forward, but can we get it? And the answer to that is absolutely, we can get these microbiomes at an industrial scale. So a lab desktop, 100 milliliters might be a big sample. 3 ounces is a big sample at a lab benchtop, especially if you're going to do genomics work. The kind of samples we're talking about is 200 million times that size. Now, our technology itself was developed internally or invented internally, and we had ARPA, DARPA, NIH, the US Air Force, all support the initial technology for biofuels. What we now recognize is that it also could be a discovery engine to develop and find molecules that will not only impact life sciences across the board, but think about therapeutics, agrochemical, sustainability, things like that. And we'll talk about some of those opportunities. So what is it that Biosortia actually does? We don't want to be a drug company or an agrochemical company. We want to focus on getting the microbes, presenting their materials. Think about their proteins, their lipids, their peptides, their small molecules, the kind of researchable materials that could be next generation products across the entire life science segment, getting those molecules, advancing those molecules through AI, derivatization, and be in a position where we can work with others to bring them next generation solutions. So how do we actually do it? Well, we created a technology, and down here on the left, those 2 pictures you see that says scouting and harvesting. Scouting is a small scale harvest from an aquatic microbiome. And then the harvesting system is about 20 times the capacity. But at the capacity we currently have, we can take a part per million of a 350 molecular weight molecule inside of a microbiome and accumulate 50 grams in 1 hour. Now we have a vision of going to a deeper scale. And the whole idea here is that with the vast majority of life science molecules being associated with microbes in the cellular signaling capability of these molecules, that uncovering those molecules will have an opportunity to advance all of life sciences. And at the scale we're currently at, we would get a very high percentage of cellular signaling molecule. At the scale we plan to go at, there is hardly a molecule that can hide from this technology. It is truly industrial scale mining of the most valuable molecules on the planet that research has never had their hands on. Now, what we will point out is it's environmentally friendly. It's not the kind of mining that chews up a mountainside. We take a sample. So imagine if we were in the Chesapeake Bay and we took a full-scale sample right now, that would be 8 Olympic-sized swimming pools. The microbes would recover probably at about the speed we can pull them out, or even faster if we could pull it out faster. So it's a non-chemical process, it's environmentally friendly, and in our testing in pursuit of this technology, we were even approved to do it in a marine sanctuary in California under state, local, and federal approval. And we think that's saying a lot as far as showing how environmentally friendly this type of approach is. So what is it that we actually do? We scout and analyze. So when we talk about scouting and analyzing, think of the aquatic microbiome just like humans, in that all microbiomes are different. And our variability of habitats is extreme. We could be where there's low oxygen. We could be where there's a high pH. So pH, oxygen, sunlight, nutrient content, salt content, all sorts of variables exist in these different microbiome. And because of that, they produce different small molecules or other products that could be interesting to researchers. So we select a site, we get those approvals and permits. Right now, our recovery rate is 20 million liters. We preserve rapidly and pulling them out without lysing cells. Then we go through our processing steps of the biomass. We lyophilize, we extract, we fractionate. That's to separate out proteins from lipids from small molecules. Then we do analysis, and then we have 5 distinct ways through the balance of our process to use AI in order to prioritize, curate, and advance these molecules in a variety of ways. With partners, we end up ultimately screening them, looking for the hits, and then advancing that IP. So Biosortia will be a IP generator that potentially within 3 years after we start full-scale execution could be one of the top 10 IP producers in the life science world because of the wealth of the capability to find molecules. Now, when we talk about the depth we are currently and where we're going, we're talking about not twice as good or 10 times as good as existing technology. We're talking about million times better than existing technology, 7 orders of magnitude better than what has ever existed when it comes to finding a small molecule in an uncultivable microbe, being able to identify it and use it. And that's key because in one execution of this technology, we will find 70 to 80,000 novel addressable molecules in order to move forward. Now, I'm going to pause there because that's a real quick overview of the technologies, a real quick overview of why these technologies really matter. And before I jump into some of the possibilities for what is the chemistry and the warfighter advantages and energy opportunities, I want to take a second in order to answer any questions, and then we'll jump into the ideation around how just in these 3 areas this technology could have an impact. And I think you have to unmute and feel free to ask a question.
Apologies if this was already asked or covered as I arrived late, but I'm curious about the effects of microplastics on your operation.
Depending upon the size of microplastics, odds are they will be collected. But when we put the water back, we won't be putting them back. So anything that is larger than 0.02 microns and is smaller than 100 microns will get captured by the system. And that's how we capture the microbiome and the viruses. So if something is within that size range, we will capture it. As our processing occurs and our extractions and fractionations, obviously, depending upon the type of plastic, it may be impacted, but likely is it will be part of what is our protein. It'll show up in our protein biomass, even though it's not a protein, when we process it through our system. It is possible, depending on how active a molecule is and how aggressive our extraction technologies are, that we could pull apart a molecule during our extraction processes as well.
Do you think that the increase overall in the water will cause difficulties in your process, or do you think it's manageable?
I think from what we've already seen, it's highly manageable. So we've done this work. We've done this work at, we call it subscale, but it's still at a scale well beyond anybody in the world has ever conceived possible. And we did not find anything that interrupted our process. And we did find 80,000 80,000 molecules that were addressable, with 73,000 of those completely unknown that came from the microbiome.
Thank you.
Any other questions on the technology before we start talking about just some of the possibilities of advancing a wide variety of technologies? So one of the things that we're going to talk about ideation over the next several opportunities here is that there are a lot of problems within the life sciences world that go unaddressed. Give you one example we're going to talk about during the environmental and sustainability side of the equation in March, and that is, think about red tide. How would a technology like this be able to solve the red tide issue? Well, if you can fingerprint the molecules of a red tide before, during, and after a red tide, you're likely to find the kind of molecules that could potentially inhibit a red tide, and that could potentially be the basis of a type of herbicide to eliminate a red tide as the bloom starts. That would be one example on the environmental side. When you talk about what we're looking at here, and we look at possibilities, future material possibilities from microbial chemistry, one of the things showed up just recently as a news article, I think it was last week or the week before, where it talked about synthetic spider silk being made by microbes. So when you talk about the material aspect of it, you could think of a wide range of different materials. That would be a possibility from this being made by microbes, or if you can get the enzymes themselves, which, remember, nature does things in a complicated way, but if you could get the base chemistry, you would be able to turn things like this into an industrial process. So it could extend not only to various fibers that nature makes, but you could advance those fibers. You could even go to something like number one in this, which is superconducting polymers made through the processes that would be well understood from nature itself. Potentially self-assembling structures, dynamic membranes that could ultimately be manufactured to have multi-purpose and be responsive. Those are the kind of things that are possible. When you talk about antimicrobial surfaces, certainly there are ways to do that, but there are so many compounds nature makes that we have no access to, that this would open up the world of chemistry dramatically. Same thing with something like self-healing polymers. Using the examples of nature and having the base chemistry of nature, you might actually be able to advance that side of material opportunities. That's just an example. Does anybody see anything on this list? Hopefully you can see this list that you would like to talk about or ask about itself. I think one of the things Eugene mentioned was anti-fouling surfaces. When you look at what nature has done, nature has perfected the ability to not have a surface end up under microbial attack. This is an opportunity that could come from the chemistry of microbes.
Okay, so everybody, please chime in. Nothing is off the table. The idea is to get the conversation going. We want to hear your ideas, your thoughts, and we can bounce them around and come up with the solutions.
Now, one of the things we'll mention. Oh, please go ahead.
This isn't on the list, but I do have a question about potential IP risks, because these are microbes, what is the risk of someone being able to take those microbes and basically have them create more microbes themselves? I'm just thinking of some industrial espionage or other sort of concerns from foreign powers.
Well, you know, when you talk about biohazards and biothreats, understanding the chemistry of microbes is imperative. Right now, the technology we're talking about is not even taught in any university anywhere in the world, let alone practiced by anybody. So this is a disruptive technology. And when you bring disruption to life sciences where nobody has been taught it, it's very hard to move it forward. So that's one of the challenges. But fingerprinting habitats with their chemistry, their small molecule chemistry, or the metabolites will be a significant way to predict what's going on with the metabolic pathways and the genes. So this is defensive, understanding these things, because you're right, the threats can come from this as well, but understanding how to deal with those threats is even more imperative. And uncovering the secrets of the dark matter of microbiology and biology opens up a big can of worms and a big can of opportunities. Right now, nobody is doing this in the world. We're trying to move this thing forward. We're too small of a company to do it on our own. We need partners, we need collaborators, we need people to work with. And that's why we're glad to be able to talk about where we're at in this technology and what it could potentially mean. But you're dead on when you say this could be a threat. Absolutely, there are molecules that we do not know about right now that are already threats to us, but we don't even know it. And this will be the way to ultimately uncover what could be those threats, how to defend against them, how to, more importantly, identify them early. That's key.
So Ross, this is Eugene. I read an article yesterday or today about a 15,000-year-old virus in the Himalayas. Is it possible to capture Antarctic ice or Arctic ice or Himalayan snow and figure out if there are things in there that could be a threat?
It is. One of the things that we've already seen and realized from our deep dive into the microbiomes that exist here is we found, believe it or not, species of microbes in Ohio that have only been associated with microbes that exist in Antarctica. So the question is, how in the world would somebody, a researcher, be able to find a microbe in Antarctica? And that's the only place it's ever been found, yet we found it in Ohio. Another example is pterin, which is a toxic molecule associated with beetles in Egypt. We found pterin in a lake in Ohio. Now, it's in very small concentrations. You've got to remember, we see what is invisible to analytical equipment because we scale it up, we amplify it so much, we can see things that are there that even the EPA can't see, let alone identify. And that's because we're going to a scale that's unheard of. We're not taking a 1 liter sample, we're taking a 1,000,000 liter sample, essentially. So there is the reality that molecules exist worldwide. There is the reality that species exist. One of the confounding things about genomics is many metabolic pathways exist in multiple organisms. So if you're defending against a particular microbe, nothing stops that metabolic pathway from horizontally gene transferring or potentially nefariously being transferred to another organism. You're looking for one organism, and all of a sudden it's producing a metabolite in a completely different organism. That's a threat that's unseen. That's how come fingerprinting metabolites, fingerprinting the metabolic pathways is part of what's really going to matter in the future, because microbes have overlapping metabolic pathways, and that redundancy in nature is typical. The least common denominator is the molecules, not the genomics. Genomics is an infinite set of possibilities, and it can create an infinite set of results. But nature, through evolution, has selected the molecules that work, and it retains them and it passes them on from generation to generation, and it also passes them on horizontally. So it's imperative that we as humanity start to focus on what we actually can't obtain right now. But we're here to say, yes, it can be obtained and it needs to get obtained. And quite frankly, with it not even being taught in universities, how in the world do we expect to advance a technology that the most storied researchers in the world don't even know about?
Right. And this is a challenge here too, because like Ross said, this is a brand new technology. This is not something that most people on this call have been exposed to and understand that you're all coming from, well, I guess you have a big learning curve. So at this point, I'd like to challenge everybody to share this with their peers, to look through these two slides, the previous slide that Ross had with all the applications, and think about where those applications are within your research, within the work that you do on a daily basis, with your peers, with your colleagues. And we have two more presentations from Ross and Biosortia, one on the 28th of February, and then I believe on the 27th of March, and bring those questions to those sessions. And let's see what we can do. I mean, the whole point of this is to make people aware of these new, novel, innovative ideas, technologies, concepts that are out there, so that we can come up with ideas on how they apply and where they apply, develop the innovations behind it. Because that is the goal of this forum, is once we get done here, we want to take these ideas, we want to move them to the collaboration platform, and we want to drive those forward to implementation. So that is my challenge to everybody on the call, is to go through those slides and come up with questions and ideas, and you can even email them, and we can get those out there as well.
Thank you, Eugene. I would add that we are very collaboration friendly and we are looking for partners. So don't hesitate. At the end of this deck, I've got some emails and phone numbers. We want to engage. I've been quoted and I have no problem. I'm sorry.
Can you put that slide up right now?
Yeah. I've been quoted as saying, and I have no problem in believing this, that is 10 years after this technology is executing at full scale, more new products in life science, therapeutics, agrochemical, sustainability, advanced energy, material sciences will come from the innovation this technology rains on researchers. This will open up the 99% of dark matter in biology the science has never had their hands on. This is the most important molecules that exist and the most valuable molecules that exist on this planet, and we don't know about them. We now have a way to start getting them. And one of the things I would say about any emerging technology, it's day one, meaning the ability to continually improve, the ability to advance it, the ability to scale it, the ability to bring tools to it that accelerate it, can't even begin till it starts. And this is day one.
All right. Well, thank you again. Again, that's Ross Youngs with Biosortia. You can reach out to Ross directly, or you can reach out through me. I appreciate you being here, Ross. And I got people that have reached out already.